Hi folks, it's Carlton from the Pharmacy Seeds Network. This will be part 3 of the biochemical sequence of nutrition in plants. And so today we're addressing silicon. It took me a good bit of research to filter through and find something that kind of touched on most of the points I wanted to touch on without going way too deep into depth. Below this video I will share some extra links. One specific one is from PubMed, or NCBI. That is a worldwide free research site where you can actually look up real research that's been done. And there is an excellent article there on silicon if you really want to go deep into depth. But we're just going to brush over some of the basics of it to get you a little more familiar with silicon as it applies to plants. So I'm going to read this to you. Of course, the link will be provided below as well, and I will share some other pictures in this video so you can have more of a visual perspective of things. So silicon needs of soils and crops. Silicon is a mineral present on Earth in abundance. This element makes up about 28% of mineral soil by weight. Until recently, this ubiquitous element was not given much attention as a possible limiting factor in soil fertility and crop production. Agronomists now recognize the valuable functions of silicon nutrition in crops and soils, and even animal life. Research conducted on New Jersey soils and in many places around the world have shown that applying supplemental silicon in a chemically available form can protect plant health and benefit crop production. Chemical names and terminology. We're going to skip that section. If you're interested in the chemistry of that, of course you can read that yourself, but I don't want to drag people down into extreme detail. So we're going to jump over to function of silicon in plants. Silicon is beneficial to many crops when added to some soils as a fertilizer. It is not classified as an essential element for most plants, but horsetail, equisetum, and some types of algae cannot survive without a supply of silicon from the environment. Many plant species, especially grasses, can take up silicon in amounts comparable to macronutrients. This high concentration of silicon in the plant contributes to plant mechanical strength. Besides a structural role, silicon may protect plants from insect attack, disease, and environmental stress by improving the plant's defense response. For some crops, silicon fertilization of soils increases crop yield even under favorable growing conditions and in the absence of disease. Specific benefits observed due to silicon nutrition are extensive. Direct stimulation of growth and yield, counteracts negative effects of excess nitrogen nutrition, suppression of some plant diseases caused by bacteria and fungi, especially Phytophthora. Diseases suppressed include powdery mildews on cucumber, pumpkin, wheat, and barley, and gray leaf spot on perennial ryegrass, and actually in most plants because it accelerates the whole biochemical process and therefore has a positive effect on photosynthesis. Suppression of stem borers, leaf spider mites, and various hoppers. Alleviates various environmental stresses. Silicon protects against lodging, drought, temperature extremes, freezing, ultraviolet irradiation, chemical stresses including salt, heavy metals, and nutrient imbalances. In animals, silicon strengthens bones and connective tissue. Vegetables, grains, and fermented grain products are sources of silicon in human nutrition. Silicon deficiency symptoms. Symptoms of silicon deficiency are generally not visually apparent in any obvious way in the field. Indirectly, silicon deficiency may be exhibited as an increase in susceptibility to certain plant diseases. Crops such as pumpkin, cucumber, wheat, and Kentucky bluegrass are susceptible to a disease called powdery mildew. Providing enhanced levels of silicon nutrition for these crops may suppress or delay the onset of the disease. When crops exhibit a high level of susceptibility to powdery mildew, this may be considered as a sign of silicon deficiency. And again, I'll point you out to, you should really do a plant sap analysis test to determine if silicon levels are up to where they should be. As a result of increasing silicon concentrations in plant tissues, the mechanical strength may be increased, helping to protect the plant from infection. Studies have shown that the amount of insect attack on plant diseases may also be inversely related to silicon uptake. So, the response of plants against pathogens and insects at the biochemical, physical, and molecular levels are remarkably similar when silicon is taken up by roots and translocated to shoots, suggesting an active role played by this element in plant defense. Grain crops lacking adequate silicon are more susceptible to lodging. If you don't know what lodging is, that's where the crops kind of fall over and like fall on top of each other, and they form these like laid over, basically what looks like lodges. That's why it's called lodging. Soil analysis. The total elemental analysis of typical mineral soil is about 28% silicon and 47% oxygen. The majority of this silicon is bonded with oxygen and other elements in the crystalline fabric of mineral soil. With weathering in time, some of this vast supply of silicon is released in soluble forms, available for uptake by plants. And I'll add to that that if you have the right soil biology, a lot more of that silicon can be mobilized directly to plants. And as plants send exudates down and feed those microbial communities will respond by making more silicon accessible. Although soil testing for silicon availability is not a routine part of soil fertility testing, some laboratories offer an acetic acid extractable soil silicon analysis. At present, the database is very limited in correlating silicon soil levels with plant silicon uptake. More researchers needed to find better soil test methods to predict silicon availability. Interpretation of any soil test requires years of field research. When 18 New Jersey soils were collected from across the state and tested using the acetic acid soil extraction method, they exhibited ranges of soil test silicon from 4 to 35 milligrams per liter, with the average soil test silicon level being 14 milligrams per liter. In our research field, some crops susceptible to powdery mildew benefited from the silicon fertilization when the soil tested greater than 40 milligrams per liter silicon acetic acid extract test. These field trials suggest that many New Jersey soils have less than optimum levels of silicon for protecting crops normally susceptible to powdery mildew. And again, I'll add, foliar feeds are a great way to supplement that directly and with much less material needed to achieve the same effect. Soil pH measurement and tests for lime requirement are useful guides for determining how much of certain types of silicon fertilizer products may be applied to a given field soil. Information on using the soil test will be discussed in the section under application rates. Factors affecting silicon availability. Soil texture refers to percent sand, silt, and clay particles in a soil. Silicon is a component of these mineral particles of varying size. Although sand is largely comprised of silicon dioxide, this material provides very little soluble or plant available silicon, and is not unusual for crops grown on sandy soils to benefit from applications of soluble silicon. And, or I'll add again, biology can help release that. In general, older and more highly weathered soils are more depleted of silicon than geologically young soils. Many of New Jersey soils are classified as ultisoils. These soils, which have been subjected to leaching in a humid environment for a very long time, tend to have less remaining weatherable minerals. Consequently, utasols tend to be relatively depleted of silicon. Silicon is not a major component of soil organic matter. Soils composed almost entirely of humus and organic matter are called muck soils or histosols. Because the substates of such soils are almost devoid of minerals, they are inherently low in silicon content. The use of peat-based soilless mixes in greenhouse production means that very little silicon is being supplied from the growth medium. Greenhouse production systems have also been shown to benefit from silicon fertilization. Some commercial greenhouse mixes are now pre-amended with silicon fertilizer. Silicon availability does not change markedly across the soil pH spectrum used to grow crops. Many of the commonly used silicon fertilizer materials also serve as liming agents, and their application results in a neutralization of soil acidity. Soils amended with plant-available silicon fertilizers will typically enhance uptake of silicon by crops for a period of several years. Thus, growers may plan a rotation cycle where a series of responsive crops can take advantage of the potential residual benefit of previously applied silicon. In summary, field research suggests that many New Jersey soils have less than optimum levels of available silicon for crop production, especially plants most susceptible to powdery mildew disease. Plant tissue analysis and crop uptake. Silicon concentrations in plants, in some cases, may reach levels comparable to or above those for macronutrients, nitrogen, phosphorus, or potassium. Concentrations of silicon in plant tissue can vary widely depending on plant species, silicon availability from the soil. Grasses and monocots in general tend to accumulate silicon. Concentrations as high as 10% silicon are possible in some plant species such as Equisteum. Concentrations near 1% are common among grasses. Equisetum, and that again is horsetail. Dicotyledonous plant species in general have fewer tendencies to accumulate silicon, and some species may grow adequately with levels of about 0.1% silicon in plant tissue. Optimum silicon concentration levels have not been established for many crops grown in New Jersey. However, research conducted on local soils and crops suggests that the concentration ranges may occur for some crops. For example, supplying supplemental silicon to a Quakertown soil used to grow pumpkin, corn, and wheat resulted in large increases in concentration of silicon in the plant tissue. Silicon concentrations in pumpkin leaf tissue increased from 700 parts per million to 3,500 parts per million. In corn stem tissue from 1,300 parts per million to 3,300 parts per million. Wheat flag leaves from 1,530 parts per million to 11,750 parts per million. And Kentucky bluegrass leaves from 4,200 parts per million to 7,200 parts per million. For optimum disease suppression and grain yield of wheat, a silicon concentration of 1%, 10,000 parts per million or more, in a flag leaf is recommended. The harvest of wheat straw can uptake and remove from soil is about 40 pounds per acre of silicon. And I will say this regarding plant sap analysis. In order to get an accurate amount of silicon available, silicon in plant material, you could do that with the dry analysis. But the downside is that you don't get to see what all the actual nutrients in the plant sap are, and so you're not getting a complete picture. This is why, again, I'll just remind you that plant sap analysis is really the great way to see what's actually going on in the plant. It's the equivalent of taking, part of a dead person and analyzing that tissue, or taking a live blood sample and looking at that live blood sample. There's a huge difference in the understanding of the nutrition that's available in that sap at that time. Crops' responsiveness to silicon. Crops may benefit from disease suppression, reduced injury from insect pests, stronger stems, and tolerance to stress or direct stimulation of yield from supplemental silicon applications. Around the world, rice and sugarcane are the crops that are well known to exhibit beneficial responses to silicon fertilization. Of crops commonly grown in New Jersey, pumpkin, corn, wheat, oats, Kentucky bluegrass, and dogwood may benefit from silicon fertilization. Crop groups that are considered good candidates for silicon fertilization include cucurbits, grasses, and small grains. Any crop susceptible to powdery mildew and or gray leaf spot would appear to be good candidates for field responses to silicon fertilization. It pretty much applies across the board, and obviously, like they say here, some plants need more than others, but having it available is part of that biochemical sequence, and it is more important than is currently recognized, or at least has been recognized in the past. More from soil rather than foliar applications of silicon. This is because of the supply of silicon to plant roots must be continually present, or it will be less effective at disease suppression. And there is a good point there about that. Foliar feeds are a great way to supplement. Ideally, what you want to do is supplement and balance your soil. Make sure there is a very active and healthy microbial community to make sure that those nutrients are made available to plants through that microbial community and in the form of microbial metabolites. And then as a foliar feed, you can really boost the capacity. It's a great way for a short-term adjustment to the plants, and that gives the soil biology time to play catch up and make those things available through the root system as well. So you're always trying to work with the system as a whole. And they go on about other stuff here, but I don't want to make this video too awful long. I just kind of wanted to introduce silicon and that sort of thing. Okay, so that's a basic introduction on silicon as it applies to plants. We covered some of the basic stuff that I wanted to cover. Went a little more in depth than I wanted to do specifically for this video, but I'd rather give you a little more info than a little less. And again, I'll share links to this and also to that big PubMed document, which is excellent and very in depth and cites lots and lots and lots of research. Well worth checking out. I hope you found this informative or educational, and I thank you for watching the Pharmacy Seeds Network, and I hope we'll see you in the next video.